Eduardo, Rocha. Go ahead.
Thank you. So thank you, the organizers, for inviting me here in this very varied and interdisciplinary forum. And today I'm going to present the work we do in our lab to try to understand how bacteria evolve by acquiring genes and functions from other bacteria. And I will focus on two questions. First is how the mechanisms of exchange that modify or diversify the kinds of exchanges that can take place. And the second one is whether this horizontal transfer of genes actually leads just to the spread of a function, or it can spur functional innovation. So the problem is now fairly well known. In bacteria, you can have a very large diversity of phenotypes within a species. Here I'm showing you E. coli. It's the best studied model in microbiology. It's typically commensal. Each of us has about 1 trillion E. coli in the gut. But some of the strains are actually pathogens. They have very different pathotypes and they have very different efficiencies. So for instance, in certain pathotypes of E. coli, you have to uptake 10 to the 8, 10 to the 9 cells to become sick with 50% probability. Whereas in other strains, it's enough to take between 10 and 100 cells to become sick with 50% probability. So you see, this corresponds really to dramatic changes in physiology and lifestyle of the bacteria that are mostly driven by horizontal gene transfer. And in species like E. coli and Klebsiella, which will be the focus of the first part of my talk, these processes of transfer take place mostly by mobile genetic elements, and typically by two of them. The first one are phages, or bacterial viruses, which can transfer DNA from one cell to the other in two major ways. Either when they package DNA into the virions, they make a mistake, and they integrate bacterial DNA, and then they will infect other cells no longer with the phage DNA, but with the bacterial DNA. This is called transduction. Or when the phage itself integrates the bacterial genome, and if it expresses traits like virulence factors or whatever, it leads to what it's called lysogenic conversion. That is, the bacteria has indeed endured a phenotypic shift that will be able to do things it couldn't do before. Now, virions are typically small, and so they typically transduction and lysogenic conversion concerns stretches of DNA up to 50, 70 kb in entire bacteria, although larger genomes are also known. Conjugation is fairly different. There's also a mobile genetic element, for instance, a plasmid, that is going to produce a bridge or a pilus between two cells. So this requires a intimate cell-to-cell contact. And then there will be machinery, a type 4 distribution system that will be able to transfer single-stranded DNA from one cell into the other. And at the end of the process, the mobile element is existing in two cells, in the donor and in the recipient. Conjugation requires cell-to-cell contact, which is not the case of transduction, but it can transfer vast amounts of information. One can conjugate the entire E. coli chromosome from one cell into the other in about 1 hour and a half. So this allows for a massive horizontal gene transfer in just one go. So once again, my talk will be in the first part trying to understand how the vehicles of transfer and the physiological state of the cell will affect the type of genetic information that is transferred and the amount that is transferred. And in the second part, then I will go into the question whether this transfer is just spreading new things that are invented by bacteria or actually spurs functional innovation. So the model we're using now at the lab is Klebsiella. This is a bacteria, an enterobacterium that's not so far away from E. coli. Well, not so far given bacterial evolution standards, and it's represented here in the picture. And you see that one characteristic of Klebsiella that's a very important one is that it's surrounded by a capsule. It is a thick layer of polysaccharides around the cell, which protects it against many things, including antibiotics, certain types of stress, or the immune system. And it's composed of a polysaccharide. And it's the first thing that these mobile genetic elements, when they're in their vehicle of transfer, pilus or phage, will face when coming into a recipient cell. So the capsule is produced by a biosynthetic pathway that is fairly well described in Klebsiella. There's a bunch of enzymes that are going to add different sugars in the polysaccharide and a mechanism to transfer to the outside of the cell where it will stay. And these sugars, that part of the polysaccharide, change very frequently, and they give rise to what is called the serotype. So a serotype is a given type of capsule with a given chemical composition. In this analysis, what we did was to collect 4,000 genomes of Klebsiella pneumonia, and this here you can find the phylogenetic tree of the species. The structure of the tree is not very important. What I would like you to focus is the ribbon around the tree. And the ribbon tells you the distribution of serotypes. I have not identified them all. There are almost 100 different serotypes in this data set. And what it shows is that it changes quite frequently. What you can probably not see, because there are too many colors, is that most of the serotypes are not monophyletic. That is, they can exist in different parts of the tree, although sometimes you have a whole chunk of strains closely related in the tree that do have the same serotype. So the goal of our project was to put together phylogeny, including phylogenetic reconstruction, ancestral trait reconstruction, recombination detection, comparative genomics, experimental evolution, and molecular genetics to understand how the fast turnover of this capsule serotype affects gene flow in Klebsiella. The first thing that set us in this path was the observation that when you take this 4,000 genomes tree and we focus on terminal branches, and most of my talk will focus on terminal branches for a number of reasons. One of them is, for instance, Eric's talk yesterday that shows you that when you come back into the tree, it's not very clear what's going on. So the first thing we did was to look at cogains, and cogains are simultaneous acquisitions, that is, acquisitions in the terminal branches in two distant parts of the tree of two genes which are essentially identical or extremely similar. Okay, so this suggests that there is some gene flow between these two lineages. And what we put forward as a question was, well, when you have cogains of identical genes in two distant parts into different terminal branches in the tree, do these branches tend to have the same serotype or do tend to have different serotypes relative to what you would expect, given the frequency of serotypes and the population structure given by the tree? What you observe is that actually there is a strong bias. So if you focus on the center graph in blue, you see that on average there is much more gene flow within bacteria of the same serotype than expected, given the data that we have. When you exclude the plasmids, so some of the elements that are involved in conjugation, you see that the bias increases even more. That is, gene flow becomes even more serotype specific. When you exclude prophages, part of the elements that are going to be driven by horizontal gene stress or driven by phages, well, when you exclude prophages, the bias decreases. So somehow this suggests that phages are driving this intraserotype gene flow. And the graph on the right shows you the same thing in a different way. We took the prophages that we identify in the 4,000 bacterial genomes. We classed them by similarity of gene repertoires. So this is what we call prophage relatedness. And values close to 1 mean that the phages have essentially the same genes and that the genes are almost identical, almost 100% identical in sequence. And if you look at this part of the graph, you see that these very similar prophages in different bacterial genomes are systematically in bacteria of the same serotypes. So once again, giving credence to this our little model, where when you have bacteria of similar serotypes, they have the same capsule, they're recognized by the same phages, and the phages drive this intraserotype gene flow. So this was fine, and we went to the lab and we wondered, okay, so now if we induce the prophages in this bacteria, how many other strains and what types of strains can they infect? And this is what I'm showing you in my left graph. It's the interaction matrix between the prophages in the bacteria, which is called the lysate, and the other strains in the analysis. And here we have 35 strains of Klebsiella. Now, the first thing that you can see in the graph is that when we induce these phages with mitomycin C and we collect them and we challenge the other strains with these phages, most of the times nothing happens. And this is why there's lots of white in my matrix. Contrary to other interaction matrices in the literature, in our case, most of the cases, we don't have interactions. Yet, there are a few cases you see that you find interactions and they're very strong. Typically, they are in blue and they correspond to interactions, positive interactions, that is the ability of the phages to infect the bacteria. It is associated when they infect bacteria of the same serotype. So it's yellow, it's different serotypes between the donor of the prophages and the recipient of the prophages. And in blue, it's the same serotype. But there are relatively few comparisons in this graph of bacteria of the same serotype. There's only about 3 or 4% of the interactions that correspond to that, even though these successful interactions tend to correspond more than a third of all of them. That is, there is a high overrepresentation of successful infections when prophages from bacteria of one serotype go on and infect bacteria of the same serotype. So once again, confirming the analysis that we had done in the genomes. So this suggests that somehow the phages recognize the capsule and use it to adsorb to the cell and to infect the cell. So this was actually known for a number of phages in Klebsiella. And we decided to do an experimental evolution to understand what was the rate of loss of the capsule when there was strong phage pressure. That is, if the capsule allows the phages to infect the bacteria, and the phages very often kill the bacteria, one would expect that if we put ourselves in a situation where the capsule is not selected for, and we add phages, then the capsule should be lost very quickly. So this is the graph on the right. We have the percentage of capsulated bacteria in our experiments, the day that passed. And if you put the bacteria in LB, which is a very rich medium, we know that the capsule is not adaptive and it tends to be lost at day 9. About 20% of the individuals in the population still have a capsule. Now, when we do the same experiment, but add mitomycin C, we induce prophages, and therefore we create a strong selective pressure of the phages onto the bacteria. And what you observe is that at day 2 or 3, there are essentially very, very few bacteria that have left or remain in the population with a capsule. As a control, if you add citrate, which is something that it's known to inhibit phage infection, you see the other way around. That is, the capsule is lost slower than under simple growth under LB. So what it suggests is that indeed phage pressure can be sufficiently strong to lead to selection of capsule loss in bacteria. Because when the bacteria lose the capsule, they eventually become almost immune to a large number of the phages that were previously capable of infecting it. So how is the capsule lost? Well, quite funny, it seems to be under the action of very strong epistatic interactions. So we look into our genomes and we select the clones where we knew the capsule was inactivated because we found inactivating mutations in the few genes that are essential for capsule production. And then we counted them and we tried to understand if there was an association between the frequency of these pseudogenes and the position of the protein encoded by these genes in the biosynthetic pathway of the capsule. And actually, there's a very nice correlation. Genes early in the biosynthesis pathway of the capsule tend to be lost first, and the genes that are involved in the later stage of the biosynthetic pathway, which is to export the polysaccharide to the outside of the cell, are very rarely pseudogenized first. And this is because, and it's now becoming clear, there are a few papers that have been come out of this in the last year. Actually, there are toxic intermediates. If the capsule is produced and it's not exported, this will eventually be deleterious for the cell. So there's sort of an order of the loss of genes under phage pressure that we observe in the genomes of the natural populations. So now we can go back to our evolutionary experiment and see if in the lab we observe the same thing. So if you remember my previous slide, we have this small evolutionary experiment where we have bacteria that lose the capsule very fast. And I forgot to say this in the previous slide, but you only see a line in these graphs, but they're actually an average of several independent experiments. The differences between the line are very significant. And so we took this experiment where the capsule is lost and sequenced the clones and tried to see which kind of genes were lost and led to the phenotypic change we observed, which is the loss of the capsule. And as in the population genetic data, we observe a very strong overrepresentation of inactivation of the genes that are very early on in the biosynthetic pathway. So this is once again a classical rule that seems to be a classical rule on pseudogenization, is that you really shouldn't start processes that you can finish, especially when they lead to intermediates, which can be toxic to the cell. So now we know that the capsule can be lost, certainly for several reasons. In the natural population, 3 to 5% of the cells don't have a capsule. What happens when the capsule is lost? So we focused on two major events. So once again, we're still looking at terminal branches. And in one of the events is the loss of the capsule, right? So you have a small tree like this, everyone has the same serotype, and then you have one terminal branch where the capsule was lost. And then you have another event, a scenario where the capsule was not just lost, it was also regained, and there is what we call a capsule swap. So we have all the pangenome, and we can infer in each branch how many genes are coming in and out by horizontal gene transfer and gene deletion. And this is what you see in my graph, how many genes are gained in overall tree, in the average genome, in the average branches which have not endured neither an activation or a swap. in the inactivated branches where the capsule is inactivated and the swapped ones. And you see that there is much more gene flow when the capsule is inactivated or swapped than the other cases. And this is interesting because if you remember my previous slides, if you inactivate the capsule, you become immune to lots of phages. So this suggests that somehow it's probably conjugation that is leading this process of capsule acquisition and the process of an excess of horizontal gene transfer when the capsule is lost. So what happens when the capsule is swapped? We took the 25 cases where the serotype was swapped in the terminal branch, and now we're comparing two genomes which are very, very closely related, the sister clade, which is still a capsule, and the focal terminal branch where the capsule was swapped. So these genomes are very similar, and you can easily identify recombination tracts. The results are here. For 24 of the 25 clones where we had a swap, we could identify a clear, large recombination tract. So the capsule locus is indicated in black in my figure, and I center my figure in the capsule locus. And you see that the recombination tracts are sometimes about 20 to 50 kb, but often they're much larger, and some of the very large recombination tracts are more than 300 kb. So this means that in one single recombination event, 5%, more than 5% of the genome was swapped by DNA that come from the outside. And this gives us a mechanism for the reacquisition of the capsule. And it strongly suggests that the vector of the acquisition of the capsule, the horizontal transfer that leads to the acquisition of the capsule, is conjugation, because if you may remember my first slides, virions typically transfer smaller amounts of DNA than conjugative systems. So is this true? Well, then we stop the lab and come back to the 4,000 genomes, and we cluster all the conjugative systems and we cluster all the prophages into families which are extremely similar. And then we, for each family of mobile element, we pose the question where do we find these mobile elements more frequently clustered within one or a few serotypes, or are they randomly distributed in the tree? So this is what you're observing here in the graph. You can see that prophages, as we expected, there is a significant bias. The prophages, which are very similar, tend to concentrate within a few serotypes or one single serotype, whereas the conjugative systems are more or less randomly distributed in the tree. So it seems that from the point of view of conjugative systems, they can cut across any kind of serotype and spread across the species, contrary to the phages. But what happens when there is no capsule? Well, to do this, we had to go to the lab, and we took 4 strains of Klebsiella of different serotypes, and we inactivated the capsule. So we have now for each strain, we have 2 variants, the wild type with the capsule and the non-capsulated mutant. And then we just test how well they receive a large plasmid, conjugative plasmid from E. coli that carries an antibiotic resistance gene. And what to observe is that systematically, the bacterium that loses the capsule, it's a better receiver of a conjugative system. That is, it's in a sense hypersexual, it receives a lot of DNA by conjugation. And this is interesting because if the capsule is acquired by conjugation, it is exactly when you lose the capsule that the bacteria becomes permissive to conjugation. And therefore, there's sort of a feedback in the sense that the probability, if you lose the capsule, it's much more likely that you'll get a new one because the bacteria becomes much more permissive to the mobile element that is likely to bring the capsule into the genome, which is the conjugative element. So our view of the world now is here. You have bacteria with a given serotype, a given type of capsule. And within the serotype, there's lots of gene flow by phages that produce within the species models of high gene flow within serotypes and low gene flow between serotypes. And then you have the conjugative elements that seem to cut across serotypes and therefore be probably much better at linking gene flow between different models of serotypes within the species and may lead eventually to the capsule reacquisition by homologous recombination. So the point here is that not only there's a lot of flux of genetic material in bacteria, but the vehicles of transfer and the ecological circumstance, physiological state of the cell when the transfer takes place are very important to understand the flow of gene information, of genetic information within the species. So it is very well known that horizontal transfer is very efficient at transferring traits that have been invented by different bacteria. It is less clear if it's also pertinent to lead to new traits, to novel functions. And I'm going to be fairly brief about this, but we have now produced a number of cases where the tools that are used for horizontal gene transfer are actually co-opted, tinkered by the bacteria to become something else and lead to new functions. So one case is the type 4 filaments, which in bacteria can serve for many things, from aggregation to motility, from natural transformation to protein secretion. And we have recently shown how these elements have diverse history of time, how from a small number of components, different organizations of the components, different number of components, and mutations that have accumulated in these components have led to very different functions in the tree of life. And our best guess is that the ancestral of all of this was involved in natural transformation, which suggests that the very mechanism of natural transformation was co-opted in some bacteria to become something like, for instance, in type 2 secretion system, a protein secretion system that will transfer big proteins from the inside of the cell to the outside. But where we find more events of co-option is actually in the mobile genetic elements themselves. And this, in a way, it's sort of expected because the bacteria are constantly flowed by mobile genetic elements that are coming all the time into the bacteria. Most of them are neutral or deleterious. And this information will accumulate, may eventually mutate and recombine. And sometimes, probably rarely, but sometimes it will lead to new functions that are adaptive and will be selected for in the population. So one example is the conjunctive pillars itself. So what you see here in my drawing is the conjunctive apparatus, which basically transfers a protein, the relaxase, and single-stranded DNA from one cell into the other. Now, it is very simple, in a sense, to go, there are not that many mutational steps to go from a conjugative apparatus to a protein secretion system that instead of secreting a protein attached to a single-stranded DNA, will just secrete another type of protein from one cell into the other. And this actually happened several times in the history of life. If you see here on the right, I have showed you the phylogenetic tree of the NPFT, which is the most abundant type of conjugative pillars that we observe in bacteria. In the ribbon, black and white represents if the element is in the plasmid or in the chromosome. Most of these elements seem to be involved in conjugation, but the branches I colored in red actually do not have a relaxase and seem to be involved in protein secretion. In some cases, this has been already demonstrated experimentally. In other cases, it's just an hypothesis, but it shows you that because this invention seems to be simple and because the bacteria keep being flooded with these mobile genetic elements, sometimes and frequently in this case, and independently in the tree, there are innovations such that the conjugative apparatus is co-opted to become a secretion system that is used by bacteria, for instance, for antagonistic interactions in pathogens or mutualistic interactions in bacterial symbionts. And so this conjugative apparatus has been co-opted in at least several protein secretion systems, DNA export mechanisms, and DNA import mechanisms. So this is for conjugation. The other key mechanism of transfer, if you remember, it's by phages. And phages also can be co-opted and seem to be frequently co-opted by bacteria. So this is a paper published a few years ago, and we traced the mutations that accumulated in prophages since they were acquired in the ancestral event of infection of the cell, right? So the mutations we are seeing and the imprint of selection on these mutations is not due to the existence of these prophages as phages in virions, but as prophages within bacterial genomes and the selective pressure they face within the bacterial genome. And we expected this to be mostly neutral, even to have many disruptive mutations so that the bacteria could get rid of the prophages that can eventually kill them. But actually what to observe is that a lot of DNA of these prophages are very rapidly lost, but some of it can be maintained for a long period of time. And when you look at that, you see very strong imprints of purified selection. That is, when you look at the ratios of non-synonymous or synonymous substitutions in these genes, there seems to be a clear tendency to counter-select mutations that are going to change dramatically these proteins, suggesting that indeed the bacteria have somehow co-opted these elements for something else. And this is what I'm showing you on the right is an example of one of these families. This is a mobile element. It was a phage. Now it's too small to be a phage. It misses essential parts of the phage. If you do the tree of the core genome of Salmonella, you get this. If you do the tree of the mobile element, you get essentially the same tree, suggesting that this has been a long process of coevolution. And we suspect these element experiments are going on. It's used by bacteria to kill other bacteria.
You
And
should
this is
try
actually,
to wrap up, maybe.
yeah, I'm going to wrap up, but just have 2 slides. And this is something that it's very common in phages. We tend to see phages as one machine, but actually they're more an ensemble of machineries that can be co-opted. They can be co-opted for defense, keeping genetic systems that repress phages, as tools to transport molecules, and they have been co-opted as toxic secretion systems where they export proteins instead of importing DNA. They can be used to dig holes in membranes for bacteria to kill other bacteria, and they are DNA packaging machineries, and they have been co-opted often by bacteria to transfer not longer phage DNA, but actually bacterial DNA from one cell into the other. And so now we've come sort of full circle because once there is a process of functional innovation, there is an individual in the population that's something new. And so we come full circle. So I started this talk by showing that mobile elements can bring traits that are adaptive to the bacteria. If this leads to phenotypic shift, this can lead to selection for this consortium of the mobile element in the bacteria. This eventually can be accommodated and improved because the function must be accommodated in the genetic background. And sometimes, probably rarely, but sometimes there's something new that comes up. And there's an individual in the population that now has a functional innovation that allows him to do something that nobody else can do. It would be probably better in most cases that this individual would keep the function to itself and grow in frequency in the population. But that's unfortunately not possible for the bacteria because very soon there will be a mobile element that will capture this new function. And now this mobile element has a new function. It will be fitter because it will be able to infect other bacteria, not only with its own genome, but with this new function that will increase the frequency of the new host. And so the cycle starts again. And once again, there will be transfer, accommodation, and eventually innovation. So this is, I'd like to thank people I'd like to thank. I'd like to thank to my lab for all the work and the people who are more specifically involved in this talk, work in Ecolab by Marie Duchamp, in Klebsiella by Olayer and Weldes, in Type 4 secretion systems by Julien Guglielmini, in Type 4 filaments by Remy Denise. In Klebsiella, the studies in bioinformatics and experimentals were mostly done by Mathieu Adiquet, in prophage domestication by Louis-Marie Bobet, and phages in Klebsiella by Georges Souza, my collaborators, and I thank you for your attention. If you have questions.
My clapping, and so thank you very much. I mean, please speak up. We don't have a lot of time for questions, but if you do, please go ahead. I have a quick question. So do you have any idea whether you can environmentally trigger or increase the rate of transfer?
Oh yeah, sure. Well, very often any environment that is stressful for the bacteria will tend to spur horizontal transfer. Both phages and plasmids tend to start the programs for transfer, so induction of the phages and conjugation in the plasmids under stress.
Okay, Anton?
Yes, I have a more general question again.
Yes.
So it seems that, well, it was known before, but the gene transfer is quite widespread. You showed that it can spread all the tree of bacteria, basically, and they can acquire some similar genes and then build similar structures and so on. You also showed, for example, that evolution in bacteria can happen very quickly. This is a loss of function, but still like capsule reduction. So under such circumstances, how can we explain the very existence of the classification, the possibility to classify actual bacteria of this structure of branching tree?
Yeah, well, I guess it's a complicated problem that I'm not going to go much into it. So it seems that there are a bunch of genes, the housekeeping genes, the core genome, if you want, that tends to be conserved more or less vertically, especially once the lineages have diverged enough that homologous recombination won't work to mix up the lineages. Okay, so within the species, as we saw yesterday, it can be very complicated. But once these genes become very divergent, they will not recombine anymore by homologous recombination because they're too divergent. The mechanism won't work. And possibly there may be a whole bunch of epistatic interactions that make that these very key functions will be not so tolerant to exchanges. And these will lead to something that is the tree of life that there is a controversy whether they can identify it or not. But I guess most people these days seem to think that we can have at least a good idea.
And I have another question. So I mean, maybe you said this in the, so if we look on long time scales for the outcome of evolution of this, you know, what the bacteria looking at, what fraction is due of the really fixed changes or changes that are significant due to horizontal gene transfer, and what fraction to more, you know, linear mutations?
That's the $1 million question. According to Henri, it's mostly transfer. Other people would say it's mostly accumulation of mutations. What is very clear is that if you take E. coli's pangenome, the diversity of gene families in the E. coli species, the average E. coli genome makes about 4,500 genes. And the pangenome of E. coli, the last time I saw it, was more than 80,000 different genes. Okay, so it's more than 15 times the average size of the genome. So most of the genes that are at low frequency in the population come by transfer. So there is no doubt on that. The doubt really stays on these core genes that tend to be kept in all the strains, whether these ones tend to evolve by mutation or by recombination. And there's the jury's out, and probably it depends on the species. Some species are much more sexually isolated than others. So it will certainly vary. But
okay, so let me change the question a little bit.
Okay.
Let's not go to the, you know, very long, let's not look at the pangenome, but let's look at the relatively long time in your lab. So
okay.
let's say the order of years, if you can. I mean, you know, something that is long by the time scale of an E. coli generation and accumulation of mutations, but I don't want to look at the pangenome and worry about all of that. I just want to get a feeling for how often does it happen, to the extent that then it gets really propagated.
We really cannot know, because the experimental evolutions that are done in the lab typically are of sexually isolated bacteria, right? They're clones, right? So you don't have exchanges. So you have no clue, right?
And they don't want to do AGT in the lab, right? They're less likely.
It's complicated, right? No, the people are starting now, but it's really complicated because well, now you can sequence and longs with longer reads, probably it will become more useful, but so far it hasn't been done very much. What we can do is using epidemiological data, right? And that we know that in a couple of decades, you can change the number of gene repertoires of genomes by up to 5%.
Okay.
In decades.
All right, we should go on. Benny, can you?
Thank you. Yeah.